Okay, it seems like everybody has got most of the stuff cut and pasted into their interactive notebooks, starting with page 40 here. Now, this next unit that we're going to be is called molecular genetics. We just did genetics, okay? We just did genetics and did dihybrid crosses. We did monohybrid crosses, codominance, pedigrees, sex-linked traits. Now we're going to take it even to a smaller level. We're going to talk about the DNA that makes the genes that those traits come from. Now, our objectives today. Number one, we're going to talk about the structure of DNA. So you better be able to, or you should be able to, tell me what DNA is made of. Okay? What DNA is made of. Also, we're going to talk about some bases that are in DNA. So you should be able to tell me the bases in DNA, and we're going to eventually get to how they match up with each other. But we're going to start off with this mighty minuscule DNA. So, the first thing on the mighty minuscule DNA is, as you can see, this guy is a blown up cell. So, on your definition, which should be the bottom of the mighty minuscule, you'll see there's a place to define the cell. What is the cell in terms of DNA? It is where the genetic information is located, and that is inside the nucleus. So if you look at this right here, these things, do those look familiar? What are those? What are those? Chromosomes. Yes, they are chromosomes. So what you should be doing right now is copying this definition of cell onto the mighty minuscule DNA sheet. Now, probably after you copy down the definition of DNA, you should probably take the cell and glue it onto the front page of the mighty minuscule DNA. You should be able to pick out a cell, I would think. So go ahead and glue that onto the front of the mighty minuscule DNA. And then you can copy down our next definition. What did we say those guys are right there? Huh? These are chromosomes. Now, it states that chromosomes are thread-like structures located inside the nucleus of animal and plant cells. Now, if you look at this, we've done this before too. This is a karyotype, a human karyotype. Okay, if you would count those, there are 22 autosome sets in this one sex set right here. So this is the karyotype of a male, because we can see the X right here and the littler Y right here. So this is a male karyotype that has the 23 pair of chromosomes that we have as human beings. So why don't you go ahead and glue the chromosome into the correct spot of the front page of the mighty minuscule DNA. These chromosomes, the second part there that I just put up, these chromosomes are made of protein and deoxyribonucleic acid, DNA. So chromosomes are made up of protein and DNA. After chromosomes, look at that cute little thing. Kind of looks like beads on a necklace. Kind of looks like beads on the necklace. That is the histone Okay, the histone is the little bead. The purple sphere there. A histone is a protein that functions as a spool for the DNA thread to wrap around. So the histone is the spool that the DNA thread wraps around. So if we look right up here, like I said, this purple sphere represents a histone. Okay? This little thread-like represents the DNA. So see how the DNA wraps around the histone. After the histone comes the double helix of DNA. So this is the thread-like part that we were just talking about. The DNA double helix is a helix or a spiral consisting of two strands of alternating phosphate and sugars. It consists of two strands of alternating phosphates and sugars. Base pairs are the rungs of this spiral ladder. Okay, the base pairs are the rungs, and they are linked together by hydrogen bonds. So if you look here, this yellow and this green, that's two base pairs. This red and this blue, that's two base pairs. So you can see yellow, green, red, blue, yellow, green, red, blue, yellow, green, red, blue. And make note here that all the yellows are with greens, all the reds are with blues. Okay, that's just not a coincidence. That is a reason for that. There is a reason that they are linked together color-wise, and we'll see that here in a bit. The final thing is the nitrogen bases of the DNA. Bases between the DNA strands form the rungs, just like I said, the rungs of this twisted ladder. The rungs of this twisted ladder. So if you look here, the A and the T, A and T go together, rung together. C and G rung together. We'll talk about that also. And here we talked about alternating sugar and phosphate. So this represents the alternating sugar and phosphate groups of DNA, and the nitrogen bases are the rungs that go together of the staircase of DNA. Now you should have all of your circles now glued into the front page of your mighty minuscule DNA. You can go ahead and color those later on if you want to. Okay? If you want to, but you can see it goes. Breaking the DNA down, it goes cell to chromosome to histone, the DNA double helix, to finally the DNA with the nitrogen bases. Now let's talk a little bit, or let's look here. I showed you just some more photos of this whole breakdown. Once again, we start at the front. Chromosome. Well, these chromosomes then break down into the histones. You see the little histones. Each one of those balls are a histone. Each sphere is a histone. And you can see that the DNA is wrapped around the histone. Here's the DNA, the double helix, and finally you can see the nitrogenous bases. Here's another picture of that. Okay, once again, the cell, the chromosome, the histone with the DNA wrapped around it, the double helix, and the nitrogen bases. Okay? Now, flip the page to page 40 in your interactive notebook. This stuff is going to start with our discussion of the structure of DNA. Now, it says at the top of that, the molecular genetic basis right there. I want you to, above that, write structure of DNA. Okay, structure of DNA. When I do these notes again, I have that changed. So instead of saying the molecular genetic makeup or model, it says, or excuse me, basics, it says structure of DNA. Now, let's talk a little bit about a genome. A genome is the complete complement of an organism's DNA. It is all of the DNA that that organism has, the genome. For many years, they've been working on something that's called the Human Genome Project, where they're trying to identify every gene on every chromosome. Okay? Cellular DNA, we know, is organized in what we call chromosomes. We just talked about those chromosomes. The DNA is wrapped around histones. The DNA is wrapped around the histones to form the chromosome. Chromosomes have what we call genes. Okay? Genes. Specific places have specific genes. If you remember, we talked about a disease called hemophilia, right? What specific chromosome was that hemophilia on? The X chromosome. Okay? You know, what else did we deal with the X chromosome? Color blindness. Okay? Those are all X chromosome genes, genes that are located on that X chromosome. Now let's talk about nucleic acids, this deoxyribonucleic acid, DNA. Now, this DNA has nucleic acids, they have bases, they have phosphates, they have deoxyribose sugar. So, one of the things that you will be doing as homework tonight is DNA, show me what you're made of, which is on page 42 there. It's going to look somewhat similar to what I have right there, okay? Once you get done. It won't look exactly like that. The colors might be a little different, but it'll look similar when you get done with this. Now let's talk about the structure of DNA. DNA is a double-stranded molecule that is analogous to a spiral staircase. Anybody got a spiral staircase in their house? Don't usually have those anymore. One of my buddies, when he grew up in high school where he lived, had a spiral staircase. Okay? But the DNA, the double helix, is like a spiral staircase. The two side rails alternate deoxyribose sugar and phosphate groups. So the side rails alternate deoxyribose and sugar. So you can see them right here. Okay, the light blue is labeled the P, so that's the phosphate. The darker blue is the deoxyribose sugar. Okay, so the dark blue's the sugar, the light blue's the phosphate. The stair steps here are the bases, okay? The base pairs. As you can see, green is adenine, red is thymine. Yellow here is cytosine, G is guanine. Typically, you're going to see that we have those abbreviated A and T, C and G. AT, C, and G. Those are the steps, the bases. Now, these little dots right here in between the bases, those are hydrogen bonds. Those are hydrogen bonds that connect the two different bases together. Purine bases have double rings. Okay, purine bases have double rings. And the purines are adenine and guanine. Adenine and guanine. So if you look right here at these bases. Here's a base, here's a base. Which one of those two is the purine? Well, it's the one that has the two rings. 1, 2, the double ring. Notice that this one is just one ring. That one right there is just one ring. That's 2, so that's a purine. Look at that one. That's a purine right there. 2 rings. 2 rings right there. That's a purine. Blue one is, or blue one. The green one right here is adenine. The purple one is guanine. So purines are adenine and guanine. Pyrimidines have single rings. Like I said, here's a single ring. Here's a single ring. Here's a single ring. The purines, or excuse me, the pyrimidines are cytosine and thymine. Now, can you notice right here that a double ring always pairs with a single ring, doesn't it? Single ring, double. Double, single. Double, single. Doesn't matter which side, but a double ring, a pyrimidine, excuse me, a double ring, a purine is always paired with a single ring, which is a pyrimidine. So, purine, pyrimidine. Purine, pyrimidine. Pyrimidine, purine. Can you identify which is a purine? Which is a pyrimidine? Okay? Because you'll need to know that. You'll need to know purines and pyrimidines, the single and the doubles. Okay? Now, your homework, ladies and gentlemen, is on page 42. Make sure that you follow the directions because you have specific colors that you need to color those, and that will be graded first thing tomorrow when we come into class. Okay?